Okay, so we're diving deep today into
Hmm.
the world of cephalosporins.
All right, sounds good.
And for our listeners out there, we're all about getting to the core of things quickly, right? So we're
Absolutely.
going to break down what makes these antibiotics tick
Right.
and why they're so crucial in, battling bacteria.
Yeah.
We'll look at their structure,
Okay.
the different
Sure.
generations, and how they actually work against infections.
Right. Yeah.
But we're going to do it without getting too bogged down in all the really technical stuff.
I like it.
Think of it as a shortcut to really understanding this key class of antibiotics.
That's a good way to put it.
So you sent over some really interesting material on
I
this.
did. Yeah.
And, one thing that really jumps out is that cephalosporins are like a major player in the world of beta-lactam antibiotics.
They are. Yeah, they're right up there with penicillins.
Okay.
In fact, they're related to penicillins, but they often have this broader reach, against different bacteria.
Oh, interesting.
And they're a bit more resilient against some of those sneaky bacterial defenses.
That makes sense.
So, yeah, they're really important.
So at the heart of these drugs, right, we have this beta-lactam ring,
Exactly.
and it's fused to another six-membered ring.
That's right.
It's fascinating how just tiny tweaks
Yeah.
at specific positions on this core structure
Mm-hmm.
can lead to these different generations of cephalosporins.
You got it.
And each one has its own kind of unique abilities.
Exactly. Like they each have their specialty.
And this is where it gets really interesting, right?
Okay, I'm listening.
These generations are largely categorized by how effective they are against gram-negative bacteria.
That's a key distinction. Yeah.
So to make this clear, let's dive into some examples.
Right. Let's do it.
Let's start with the first generation.
Okay.
Drugs like cefazolin or cephalexin.
Yeah, those are the classics, the first ones on the scene.
Right.
Think of them as like the pioneers, the workhorses.
Okay.
They're really good against common gram-positive bacteria,
Uh-huh.
like your Staphylococcus and Streptococcus, those kinds that cause skin infections.
Right, right.
And they're also really important in preventing infections before surgery.
Oh, that's interesting.
But, they do have a bit of a weakness.
What's that?
They can be vulnerable to those bacterial enzymes, the ones that break down beta-lactam rings.
Like little Pac-Mans.
Yeah, exactly like that.
So that brings us to the second generation.
Okay.
Drugs like cephardicin, cefoxid, and cefotin.
So with these, we see a step up, right?
Oh, okay.
They're getting better at tackling those gram-negative bacteria while still holding their own against the gram-positives.
Right.
And some of them even go after anaerobic bacteria.
Anaerobic bacteria.
Yeah, the ones that can live without oxygen.
Oh, wow.
And they're a bit tougher against those beta-lactamase defenses.
Oh, so they're evolving?
They are. They're getting smarter.
That's incredible.
And you often see these used for respiratory infections or infections inside the abdomen.
Oh, I see.
Yeah, so they're moving into new territory.
So then we have the third generation.
Okay, this is where things get really interesting.
Right.
We've got ceftriaxone, cefotaxime, ceftazidime.
Big names.
These are the big guns. They're the broad-spectrum powerhouses,
Right.
really effective against a wide range of gram-negative bacteria,
Uh-huh.
including some pretty nasty ones.
Wow.
And get this, some of them can even cross into the brain.
Into the brain.
Yeah, so they're vital for treating really serious infections like meningitis.
Oh, that's incredible.
And you mentioned ceftazidime. That one's particularly good against Pseudomonas.
Pseudomonas.
Yeah, it's a tough one to fight.
Wow, so these are like the elite forces.
They are. They're the special ops of antibiotics.
I like that analogy.
But it's worth noting that they might be a tad less active against gram-positives compared to those first-generation drugs.
Oh, that's interesting. So there's a bit of a trade-off.
Yeah, there's always a balance, right?
Right.
You gain something, you might lose a little something else.
That makes sense.
It's all about choosing the right tool for the job.
Okay, so moving on to the fourth generation.
All right, so now we're talking about cefepime.
Cefepime.
This one's a real multitasker.
Okay.
It's got that extended spectrum, good against both gram-positives and D-gram-negative.
Wow, so it's like the best of both worlds?
Pretty much, it's like a hybrid.
So it can handle Pseudomonas too.
You bet it can. It's got the power.
That's impressive.
And it's also more resistant to some of those pesky beta-lactamases.
Oh, so it's even tougher than the previous one.
Exactly. It's like they've learned from the previous generations.
That's amazing.
So you often see this one used for those really severe infections, the ones you might pick up in a hospital.
Oh, so like the heavy hitters?
Exactly the ones you bring in when the going gets tough.
And finally, we have the fifth generation.
All right, the new kid on the block.
Okay.
This is where we see ceftaroline taking center stage.
Cephalosporins.
This one's got a very specific target in its sights.
Okay, tell me more.
It's got the ability to fight MRSA.
MRSA.
Yeah, that's methicillin-resistant Staphylococcus aureus.
I've heard of that one. It's a tough one to beat.
It is. It's a real problem, but ceftaroline is up for the challenge.
Wow. So it's like the superhero of antibiotics.
You could say that it's definitely a game changer.
That's incredible. So it's used for complicated skin infections and pneumonia.
That's right. Especially when there's a concern about MRSA being involved.
Wow, that's really important.
It is. It shows how we're constantly adapting to these evolving bacterial threats.
So we've talked about how these different cephalosporins are designed to attack bacteria.
Yeah, we've seen their evolution.
But how do they actually do it?
Well, like penicillins, they work by messing with a bacterium's ability to build its cell wall.
Oh, okay.
Think of the cell wall like a brick wall.
Okay.
And the cephalosporins are targeting the construction workers.
Oh, construction work.
Yeah, these proteins called penicillin-binding proteins, or PBPs.
PVPs.
They're essential for building that wall.
Okay.
So the cephalosporins come in and bind to these PBPs. It's like jamming their tools, you know. So they can't link those building blocks together properly.
The peptidoglycans.
Exactly. Those are the bricks.
And without a stable wall.
The bacteria can't survive. It's like a house of cards.
That's a great analogy.
They just fall apart.
So it's a pretty clever way to take them down.
It is. It's exploiting their weakness.
But of course, bacteria are tricky.
Oh, they're incredibly adaptable.
They've developed ways to resist these drugs.
They have. It's a constant arms race.
So how do they do it?
Well, the sources highlight a few key resistance mechanisms.
Okay, let's hear them.
One of the main ones is producing these enzymes called beta-lactamases.
Beta-lactamases?
Yeah, think of them like tiny molecular scissors.
Okay.
They break open that crucial beta-lactam ring in the cephalosporin molecule.
So it's like disarming the weapon.
Exactly. They render it useless.
And there are different types of these beta-lactamases.
Oh, yeah, there's a whole family of them.
Like what?
Well, you've got your penicillinases, those mainly target penicillins. Then there are cephalosporinases, like AMPC. Those go after cephalosporins specifically. And then you've got the really worrisome ones, the extended spectrum beta-lactamases, or ESBLs.
ESBLs, those sound serious.
They can even break down those powerful third-generation drugs.
Oh, wow. So they're like the elite resistance fighters.
You could say that they're the ones we're really concerned about.
So it's not just those enzymes, right?
No bacteria have other tricks up their sleeves.
Like what?
Well, they can alter their PBPs.
They're penicillin-binding proteins.
Yeah, so the cephalosporins can't bind to them as effectively.
So it's like changing the locks.
Exactly, they're making it harder for the drugs to get in.
That's sneaky.
And then in gram-negative bacteria, they have this outer membrane.
Okay.
They can make it less permeable, so it's harder for the antibiotic to even get through.
Like building a thicker wall.
Yeah, exactly. They're reinforcing their defenses.
And they can even pump the drug out of the cell.
That's right. It's called efflux.
Efflux.
They literally push the drug back out.
Wow, so they've got a whole arsenal of defenses.
They do. They're pretty resourceful.
It's amazing how they can adapt so quickly.
It's a testament to their evolutionary prowess.
So finally, let's talk about where these amazing drugs come from.
All right, so it all starts with a fungus.
A fungus.
Yeah, a fungus called Acremonium chrysogenum.
Acremonium chrysogenum.
It produces the initial core structure of cephalosporins. It's called 7-ACA.
7-ACA.
And this is all done through a process called fermentation.
Oh, like brewing beer.
Kind of similar, yeah.
That's fascinating.
And then scientists take this naturally produced 7-ACA and they make modifications in the lab. They tweak it and create all those different cephalosporin drugs that we've been talking about.
So it's like they're taking nature's blueprint and improving on it.
Exactly. They're harnessing the power of nature and refining it.
That's really cool.
It's a beautiful example of science and nature working together.
So to wrap things up, this deep dive into cephalosporins has really shown us how a relatively simple core structure can be modified to create this amazing arsenal against bacterial infections.
It's pretty remarkable, isn't it?
It is. And understanding these different generations and how bacteria develop resistance,
right,
it gives you a real appreciation for the complexity of this fight against infectious diseases.
Absolutely. It's an ongoing battle.
It is, and it's constantly evolving.
That's for sure.
What I find so fascinating is how these seemingly small chemical changes can lead to such huge differences in how these drugs work.
It's like a tiny tweak can make all the difference.
And which bacteria they can target.
It's all about precision.
Right.
Hitting the right target with the right weapon.
And it makes you wonder, right?
About what?
About this constant back and forth between us developing new antibiotics and bacteria finding ways to resist them.
It's an evolutionary arms race.
It is. And it makes you think about the future.
What about it?
What are scientists going to come up with next?
That's the big question.
As bacteria become more and more resistant,
Yeah, the stakes are getting higher.
what new approaches are we going to need to explore?
That's something for everyone to ponder.
It is food for thought.
It really is.
So for our listeners out there, think about that.
Yeah, what completely new or unexpected strategies do you think scientists might come up with to combat infections in the future?
It's a challenge, but I have faith in human ingenuity.
Me too. We'll find a way.
We always do.
We have to.
That's the spirit.
That's how we keep moving forward.
Absolutely.
Onto the next challenge.
On to the next generation
Exactly.
of antibiotics.
And of scientists.
That's a great note to end on.
I think so too.
Thanks for joining me today.
It was my pleasure.
Until next time.
See you then.